How, and when all the alleles were formed, where this occurred, and how we will apply the out-of-Africa and multi-regional theories to explain allele creation. We leverage a triple cohesive framework of comparative genomics, anthropological knowledge, and paleogenomics to integrate current known alleles. While none of the three methodologies employed in this research can be considered ideal, it is important to note that, currently, there is no methodology available that is both unique and perfectly suited to address all aspects of the research objectives. The human diverged from orangutans around 14 million years ago, from gorillas about 9 million years ago, and from chimpanzees roughly 7 million years ago. Early hominins such as Sahelanthropus, Ororan, and Ardipithecus lived between 7 and 4.4 million years ago in Central and East Africa, where the formation of lakes Tanganyika and Malawi provided essential resources within the East African Rift. Australopithecus inhabited eastern and southern Africa, where the earliest stone tools were found. With Homo habilis, major changes began, including the shift to omnivorous diets, habitual bipedalism, greater tool complexity, and a broader geographic range. This transition continued into Homo erectus, first species to leave Africa, but experienced a severe population bottleneck between 930,000 and 813,000 years ago due to extreme glaciation. During this period, the fusion that created human chromosome 2 may have contributed to reproductive isolation and speciation. Later species include Homo heidelbergensis, Neanderthals, and Denisovans. Homo sapiens, emerging about 315,000 years ago, represents the only surviving human species today. Our company conducts a comprehensive, end-to-end analysis of every step involved in the formation and diversification of human alleles. Rather than treating alleles as static entities, we model them as dynamic evolutionary outcomes shaped by time, population history, and molecular mechanisms. All extant human alleles arise through a sequential order of mutations, SOM, recombination events, or a combination of both. These processes are not random in isolation but occur within well-defined evolutionary and demographic contexts. Our platform systematically captures these processes through three integrated and complementary methodologies, which are described in detail throughout this video. To properly frame this work, it is essential to place human genetic variation within its deep evolutionary context. Our lineage diverged from that of orangutans approximately 14 million years ago, a critical evolutionary separation that establishes the temporal baseline for our ancestral reconstructions. This divergence, discussed in the video, marks the beginning of a complex series of branching events that eventually gave rise to modern humans. By anchoring our analyses within this timeframe, we are able to trace allelic origins not only within Homo sapiens, but across multiple hominin and great ape lineages. Building upon this evolutionary framework, we have undertaken large-scale protein annotation efforts to support downstream genetic and functional analyses. Using multiple scientific database platforms specializing in genetics, molecular biology and bioinformatics, we have annotated more than 20,000 human proteins, ensuring standardized identifiers, functional descriptions, and cross-database compatibility. From this broader set, we curated approximately 4,000 proteins into two strategically defined groups. The first group comprises genes with established or emerging links to human disease and is consolidated under AI genomic resources. The second group focuses on genes relevant to pharmacogenomics, PGx, which we further subdivide into pharmacokinetics and pharmacodynamics to capture both drug metabolism and drug response mechanisms. Beyond protein-level annotation, our work emphasizes the reconstruction of allelic and ancestral histories at an unprecedented level of resolution. We have fully developed annotations, ancestral frameworks, and evolutionary tracing for dozens of genes with high clinical and biological relevance, including CYP2D6, CYP2C19, NAT2, HLA-A, C, HLA-DRB1, and HLA-DQA1, among many others. These genes were selected not only for their medical importance, but also for their rich evolutionary signal, which allows robust inference across species and populations. Our analytical workflow starts with trusted reference datasets sourced from established biomedical databases. From these sources, we annotate all known human alleles and systematically reconstruct ancestral frameworks by incorporating homologous sequences from orangutans, gorillas, chimpanzees, bonobos, and additional hominins when available. This comparative approach enables us to distinguish ancestral states from derived variants and to identify lineage-specific mutational events with high confidence. Genes' evolutionary trajectory can be examined directly within our system through a standardized 6-step analytical pipeline, designed to integrate molecular, structural, population, and evolutionary data into a single coherent framework. The first step, gene list and annotation, involves the comprehensive compilation of single nucleotide variants and alleles. This is achieved through automated web scraping and data integration from UniProt, Ensemble, NCBI databases, including nucleotide, protein, and SNPs resources, PharmVar, UCSC, and other curated repositories. This step ensures completeness and traceability of variant data. The second step, gene backbone construction, represents the core integrative layer of our platform. Here, we combine allele frequencies, molecular conformations, ancestral relationships, estimated allele ages, and geographic distributions. Additional layers include color-coded lineage mapping, parent-child allele relationships, recombination events, three-dimensional structural data, and inferred migration routes associated with population expansions and contractions. Together, these elements form a multidimensional representation of gene evolution. In the third step, allele publishing, curated allelic data are released and visualized on our platform. This ensures transparency, reproducibility, and accessibility for researchers seeking to explore or validate evolutionary and functional hypotheses. The fourth step focuses on three-dimensional conformations, where structural biology is integrated into evolutionary analysis. By modeling protein structures across alleles, we assess how specific mutations alter folding, stability, or functional domains, providing a mechanistic bridge between genotype and phenotype. The fifth step addresses the sequential order of mutations, SOM, and associated SNVs. Rather than treating variants independently, we reconstruct the temporal sequence of mutational events and linked variants that gave rise to each extant allele. This approach allows us to resolve complex haplotypes and to infer causative versus other mutations. Finally, the sixth step, allele and migration pathways, maps evolutionary trajectories onto population history. By integrating genetic data with inferred migration routes, we reconstruct how alleles spread, diversified, or declined across geographic regions and time periods. To demonstrate the full scope of this pipeline, we present three representative case studies. HLA-B exemplifies a gene with exceptionally rich data extending from orangutans to modern humans, allowing fine-grained ancestral resolution. CYP2D6 illustrates allelic diversity across multiple hominin lineages, highlighting the impact of both mutation and recombination. NAT2, in contrast, showcases extensive diversity within Homo sapiens, making it an ideal model for population-specific adaptation. Together, these examples illustrate the scalability, depth, and translational relevance of our analytical framework.